Okay, everybody, we're going to go ahead and get started. Welcome for those of you in person and those of you on Zoom to one of our most exciting events that we have in the Department of Psychology, our faculty lecture series. Today, we're lucky to have Joni Wallis speaking to us, and I won't take too much time with the introduction. Just a brief bit of background for those of you who don't know Joni. She got her PhD in, well, let's just say several years ago at Cambridge University, and her PhD is in anatomy. That's a fun fact you may not have known. She went on to do a postdoc with Earl Miller at MIT, and then we were fortunate enough to recruit her here to UC Berkeley in 2004, where she's been ever since, with a split appointment between the Department of Psychology and the Helen Wills Neuroscience Institute. Her work has been continuously funded, as best I can tell, since she got here from 2005 from all sorts of federal agencies. And she publishes in remarkably, I think, in the top journals all the time. I lost count of how many Nature Neuroscience publications she has. Neuron, Journal of Neuroscience, TICS, got a Best Paper Award in TICS Trends in Cognitive Science. And her work is of great interest to not only people in neuroscience, but also cognitive science, and in my area, clinical science, because she tackles such a fundamental set of questions. How does the prefrontal cortex mediate important kinds of behaviors like decision-making, goal-directed behavior, reward processing? Her writing is beautiful, and it reaches a wide audience. That is, I think, at least from somebody outside of neuroscience, something that you don't often see. She has developed and pioneered many new methods for the work that she does. She'll probably tell us about those today, but I think it's remarkable that she created a method to record hundreds of neurons at the same time in real time to watch what's happening in the brain as decisions are being made. So a pioneer in many ways. So I will now stop talking and turn it over to Joni Wallis.
Okay, thanks, Dan. So yeah, thank you for that introduction. Can everybody hear me okay? Okay, perfect. All right, so I should just apologize at the outset. I'm on a medication at the moment, which sometimes makes me woozy and faint. So this is my emergency chair, just in case something happens. Okay, so I'm going to tell you about the research that goes on in my lab. We study this area of the brain, the orbital frontal cortex, and the role that it plays in decision making. And recently we take an approach that I call, taking a brain-machine interface approach to this problem, and I'll explain what that means as we go through the talk. Okay, so this is the part of the brain that interests me. It's basically the part of the brain that rests just on top of your eye orbits. It interests me because it's a really nice example of the localization of a cognitive function. So we know that damage to orbital frontal cortex impairs decision making, and we don't see impairments of decision making with damage to any other region, any other cortical region in the brain. And we also see it's dysfunctional in many neuropsychiatric disorders that involve an element of impaired decision making. So things like mood disorders, addiction, eating disorders, obesity, and obsessive compulsive disorder. And so just to give you a sense of what these patients are like, this is a description of patient Elliot. He's probably, after Phineas Gage, one of the most famous orbital frontal patients. So he was in his 30s when he was diagnosed with a tumor in the ventral medial part of the frontal lobe. And during the removal of that tumor, his orbital frontal cortex was damaged, and you can see that with all of this black on the MRI scan. Now, after the operation, he looked remarkably fine. So no sensory motor problems, normal language. He was of high IQ before the operation, and he was still of high IQ after the operation. Good memory and normal performance on standard tests of frontal lobe function. But his everyday life just became a disaster. So within a few months of the operation, he quit his job as an accountant in a building firm. He went into partnership with what essentially was a con artist. Everyone warned him against it, and he lost all his life savings. He divorced his wife of 17 years and remarried another woman within a few months. That woman was actually a sex worker, and that marriage only lasted a few weeks. He then went and moved back in with his parents, found a job 100 miles from their home. That job only lasted a few weeks because he was fired because he didn't show up on time. Not surprising with a 100-mile commute. So having made a series of, solid life decisions all the way through to this operation, within a few months of the operation, his life is just a disaster. And this isn't a one-off. So we see this over and over again with these patients. And you can even take them into the lab and give them a very, very simple choice. And probably the simplest example is where you hold up 2 sheets of paper and say, which color do you prefer? And normal people can do that. We have color preferences. OFC patients, they're just choosing at random, right? So even something as simple as that, they've lost the ability. Now, in terms of its involvement in neuropsychiatric disease, here's a couple of examples. This is changes in cortical volume in depressed patients. You can see that those changes are taking place in these frontal limbic regions. So the orbitofrontal cortex and the adjacent medial prefrontal cortex. And this is a series of coronal sections through the frontal lobe of a macaque, showing where the serotonin transporters are. These are a target of antidepressant medication. And you can see again that the transporters are really in the orbital and the medial regions and not in the lateral prefrontal areas. So because of this, the orbitofrontal cortex has become a target for people trying to manipulate these circuits in order to treat these neuropsychiatric conditions. Now, one method has been deep brain stimulation. And just to give you a sense of how deep brain stimulation works, we've used it for a long time now to treat Parkinson's disease. Electrodes are basically implanted in the area that you want to target. Then the leads are buried under the skin, and the battery packs are implanted in the patient's chest cavity. And this has been very successful for certain conditions. If you've not seen it before, this is a slide that Rich Ivory lent me many years ago. So here we have a Parkinson's patient, shuffling gait, tremor, stooped posture, and lack of facial expression. And then here's that same patient with the deep brain stimulator in. So you can see no stooped posture, normal gait, the tremor is gone. And when he turns around, he has a big grin on his face. So emotional expression is back. Now, when that same approach has been tried in the orbitofrontal cortex, it's been pretty disappointing. It hasn't really worked, and it's been used for treating OCD and also depression. And the clinical trials, in fact, that focused on the singular cortex were halted really early on because it was clear that they were just not, they were not being efficacious. Now, we think that the reason why this isn't working in these structures compared to how it works in the basal ganglia is probably because of the nature of the signal. So deep brain stimulation is a very crude method. It essentially relies on chronic tonic stimulation, and that probably just simply isn't that meaningful to the cortex. Now, in contrast, there have been efforts to more meaningfully interact with cortical circuits, and these have been really pioneered by the motor field. And this is the domain of brain-machine interfaces. So this has been an approach used to treat paralyzed patients who have an intact motor cortex, but no way for that motor cortex to communicate with the muscles and the skeletal system. And so the aim of a brain-machine interface is to have a brain implant that essentially records neural activity directly from the motor cortex of paralyzed patients, so that those patients can then, so that activity can then be used to control machines like wheelchairs or computers or robot arms. And here we have a monkey in which this work has all been developed. And here, in fact, is a video of a monkey. And he has an implant in its motor cortex. And with that implant, he's controlling this robot arm so that the robot arm can reach out and grab pieces of marshmallow and deliver them to his mouth. And you can see that, you know, he's got the pincer grip of the robot arm, and he can locate the arm anywhere within three-dimensional space. So the objective of our research is to try to adopt this same kind of brain-machine interface approach that's been so useful for the motor system, and to bring it in to understand cognitive processes, and ultimately with the aim of trying to develop treatments for neuropsychiatric disorders. And so the idea is, can we modify maladaptive behaviors that underlie these conditions, so things like addiction or, you know, craving, by reading out neural activity from prefrontal areas like orbital frontal cortex, and then essentially writing in adaptive behaviors through electrical stimulation. That's sort of like the you know, 50-year research program. Now, everything I'm going to tell you about is from the monkey. And this slide illustrates why. This is the cytoarchitecture of the medial and orbital frontal surfaces of the frontal lobe in the human, the monkey, and the rat. And you can see that the organization from the monkey to the human is very, very similar. In fact, this is this is one of the oldest, phylogenetically oldest areas of the frontal lobe. And there's really good conservation of the layout of the organization of these areas across primates. But you can also see how that homology breaks down when we move to the rodent. Okay, so here's the outline of what I'm going to talk about today. I'm going to tell you first of all about our efforts to read out information from orbital frontal cortex, then talk about how we can write in information, and finally talk a little bit about the sort of a new line of research that we've been doing over the last couple of years, looking at the role of the hippocampus in decision making. Okay, so how do we read out information from OFC? So this is the typical kind of experiment that we do in my lab. So we basically train the animal on this bunch of pictures that are associated with different reward outcomes. And they learn these associations really quickly. They'll learn 30 pictures like this in about a day. And you can see that this set of pictures varies in terms of the probability that you're going to get a reward. This set of pictures varies in terms of the amount of reward that you're going to get. And then this set of pictures varies in terms of the amount of work the animal's going to have to do in order to earn the reward. And once they've learned these associations, we then present them with pairs of pictures and ask them to make a choice. And so we then record the activity of orbital frontal neurons. And here is a typical orbital frontal neuron. So what you're looking at here is time along the x-axis while the animal's making his decision. And then we've got firing rate on the y-axis. The different colors of the lines indicate the value of the thing that the animal's going to choose. So you can, and this is all the activity of one neuron. You can see that the neuron's firing rate increases as the value of the thing that the animal's going to choose increases. And it's a very abstract measure of value, right? It doesn't matter whether we're physically manipulating value with effort, payoff, or probability, the neuron responds very similarly. And about half the neurons show this response, an increase in firing rate as value goes up, and about half show the other response, which is an increase in firing rate as value goes down. So you have this kind of opponent coding scheme in orbitofrontal cortex. So I should say that this slide represents really about 10 years of research in our lab, kind of amalgamated together. When we first discovered these kinds of neurons, we were very excited. You know, it potentially explained why orbitofrontal patients had a different difficulty with decision making. If you're missing the neurons that tell you the value of things in your environment, you're going to have to choose more randomly. And so we went, and a whole bunch of other labs did as well, and figured out all sorts of different ways that we could manipulate the value of things for a monkey. And basically, didn't matter what way we chose, it was always represented in the orbitofrontal cortex. But eventually you kind of hit a wall with that, and you realize that you're not really making very much progress. Because part of the problem is that we're not actually getting at the decision-making process itself. We're getting at sort of this final, the result of the decision-making process. And in fact, this is a problem for all of us studying cognition, right? Which is that we can't measure the cognitive process itself. The cognitive process is hidden from view. And so we have to infer the cognitive process via other measurements, whether they are, you know, reaction times or choices or effects on discriminability. We're always measuring something indirect and then inferring the presence of the cognitive process. You can see that here where, you know, this guy's faced with this choice and he's going to make the same choice every single time, but the cognitive process that led to that choice is different on all three occasions. Now, one way in principle that we can get around that is to record the cognitive process directly from neural activity. But there's challenges there. Firstly, it means we have to understand the neural code, which is not easy. And secondly, neurons are really, really noisy. So the standard way that we deal with that is that we do the same thing over and over again, and then we average across all of our trials, right? So here we have one of our neurons. We're presenting the reward predictive cue. The dots indicate the spikes on 40 or so different occasions as we do this. Now, obviously, there's clearly a response here, right? The neuron shows roughly the same response on each trial, but the exact pattern of spikes is very different every time. And we don't understand that noise, right? We don't understand whether it's coming from the brain, whether it's intrinsic to the neuron, whether it's intrinsic to the cognitive process. We don't know where neural noise comes from. So we average across all that noise and we construct our spike density histograms like I showed you on the previous slide. Now, one alternative approach is, rather than integrating across trials, what we can do is we can integrate across neurons. So now we are recording, this is a single trial of activity, and we're now recording across 40 neurons. And we take that pattern of activity evoked by those neurons and we project it into a high dimensional space. In fact, this example here, this is a leech. And I think the leech was engaged in two different behaviors, swimming or crawling. And you can see that the two different behaviors evoke activity patterns in two different regions of this space. So now by looking at the neuronal activity, we can determine what behavior the animal is engaged in. Now, of course, that's not that interesting because we could just look at the animal to determine what behavior is engaged in. It potentially becomes more interesting, though, when we're looking at cognitive processes, right? We could, in theory, do this, look at our neural response and decode what the cognitive process was going through. So this is an experiment that Erin Rich did when she was in my lab. She now has her own lab at Mount Sinai in New York. So she did very much a similar experiment, what I just told you about before. We now have 8 pictures that predict 4 different amounts of juice. We are now recording though from ensembles of orbitofrontal neurons between about 10 and 20 value selective neurons. And we present these pictures to the animal one at a time. And we record the pattern of activity that those pictures evoke. We then train a classifier to decode what value is being represented in the orbitofrontal cortex. So we have 4 different values. So chance performance would be about 25%. And our decoder performs at about 50% accuracy. So it's well above chance, but still, a long way off perfect. But again, that's not that interesting, right? Because we know which picture we presented to the animal. But the idea is that if I know the pattern of activity evoked when the animal's thinking about option A, and I know the pattern of activity when the animal's thinking about option B, I can now give it the choice between A and B and look at what happens in orbitofrontal cortex. In fact, what happens is shown in this slide here. So these are 6 different trials that have been chosen at random with time along the x-axis. And now on the y-axis, we have the output of the decoder, which is the probability that the neural activity resembles one of those values that the decoder has been trained upon. So there's a few things to notice. The first is it's really noisy, right? There's plenty of time when the decoder is not really sure what it's looking at in the neural activity. However, there are also periods of time when the decoder is really sure that that pattern of activity is one that it knows the value of. I should also say that we've color-coded these lines according to the different values. So blue represents the high value option that the animal's going to choose. Red represents the low value option the animal's not going to choose. And remember, there were 4 different values, but we're only ever giving them a choice between 2. So the black is the ones that were not presented. So you can also see that there's a lot of dynamics in this signal. There are times when the decoder seems to flip-flop back and forth between representing one option and then representing the other option. So you can see that most clearly here, but it's also present here and down here. Now we can also tell that these dynamics are not simply noise, right? Because you'll also notice that the options that are not available, the black options, those are rarely decoded from neural activity. Now, we were able to use these dynamics to predict the animal's choice performance. So it turns out that these flip-flops, what we call these state transitions between representing alternate options, were more common when the animal was going to choose suboptimally, when he was going to make an error and choose something less valuable. They also predicted how quickly the animal was going to make his choice. So if the decoder went and decoded the high value chosen option very quickly and very strongly, then the animal was going to choose quickly. There was a negative relationship with reaction times. In contrast, if the decoder represented the low value option very strongly, that would slow down the animal's reaction times. So we were able to both predict how optimally the animal was going to choose and how quickly it was going to choose from this decoder. Now, one thing that we couldn't understand is how these dynamics actually led to the animal making the choice response. And there are a couple of puzzles. The first thing is that the orbitofrontal cortex doesn't encode very much motor information. It's not very well connected to the motor system. But there was also no evidence that the orbitofrontal cortex needed to be encoding a particular value before the animal made his choice. In fact, the orbital frontal cortex would carry on flip-flopping backwards and forwards, even as the animal made his choice and received an outcome. One clue is the fact that there was the different valued options were not represented equally. So higher value options tended to be represented more frequently, and they also tended to be longer duration once they occurred. So that means that an area downstream of the orbitofrontal cortex that was sort of listening to this flip-flopping backwards and forwards and integrating that information could potentially use that information to determine what the optimal response would be. So we fit this using an accumulator of evidence model, and this was taken from the perceptual decision-making literature, where you're trying to decide on make decisions about sensory information. And the idea is essentially that when the orbitofrontal cortex is representing the value of the thing on the right, it would sort of push the motor system towards choosing the thing on the right. And when it's representing the value of the thing on the left, it would push the motor system towards choosing the thing on the left. And because the higher value option is represented more often and it lasts longer, that would mean that in general, the motor system would choose the higher value option. So we looked for evidence about where this would potentially occur, and this was work done by Zuzana Beluski, a graduate student in my lab who's just about to defend her thesis. She recorded from a bunch of downstream areas that are potentially downstream of the orbitofrontal cortex and that encode both value information and motor information, and so could potentially be doing this computation. I'm not going to tell you about her striatal work. She just got that accepted into Neuron, but I am going to tell you about her work in anterior cingulate cortex. So basically, she was recording now simultaneously from these two areas. And the first thing that we noticed is that in the anterior cingulate cortex, there is a ramp up in information about the choice response that peaks about 100 milliseconds before the animal actually makes its response. So that was already encouraging. This is what you would expect from an area engaged in selecting the correct motor action. And now the question was that ramp affected by what was going on in the orbitofrontal cortex? And the answer is that it was. So if we basically synchronize our neural activity now to the flip-flopping in the orbitofrontal cortex, and we look at the 100 milliseconds following the onset of one of the states in the orbitofrontal cortex, if it's the more valuable state, the anterior cingulate cortex ramp is steeper, it's faster. Whereas if the low value option is being represented, that there isn't, the ramp is shallower. So consistent basically with this idea of the anterior cingulate cortex integrating the dynamics from the orbitofrontal cortex. Okay, so let me summarize what I've told you so far then. So we've seen that decision-making involves these alternating value states in the orbitofrontal cortex. We've seen that the dynamics of those states can predict both the speed and the accuracy of the decision. We've seen evidence that the anterior cingulate cortex is listening to those dynamics in order to determine the correct choice response. And we've also seen that understanding, being able to measure the dynamics of the cognitive process in our orbitofrontal cortex has enabled us to better interpret the activity that's going on in the anterior cingulate cortex. In fact, that is potentially true of all of the areas in the brain, right? That we can now get a better handle on what those areas are doing because we know the dynamics of the decision-making process itself. Okay, so what about writing in information? So this is work that Eric Knudsen has been doing, postdoc in my lab. So very similar task. We're going to have these reward predictive cues. He's going to make a choice. And those reward predictive cues are associated with a particular probability of either getting a reward or not getting a reward. We now only actually have 3 pictures. We're presenting 2 of them on any given trial. But the difference is now that the value of those pictures isn't static. They're going to vary across the course of the trial. And you can see that here. So each of these is showing the value, the probability of getting reward associated with each of the pictures. This is the session. So you can see over here, for instance, that pink is low value, blue is middle value, and green is high value. Whereas over here, the pink is middle value, the blue is high value, and the green is low value. So there are periods where there are stable periods where the contingencies are relatively static. And then there are these drift periods where the contingencies of at least one of the pictures is changing. And the solid lines are the objective probabilities of getting a reward. The dotted lines is the probability that the animal is going to select that picture. So you can see the animal's doing a really good job of tracking these contingencies and adjusting its behavior. It's just taking him a few trials to adapt to the new contingencies. Now, when we did this, one of the things that we noticed is that there was now a very strong, very prominent theta oscillation in the neural activity in the orbital frontal cortex. So this is an oscillation between about 4 and 8 hertz. And that oscillation was synchronized to salient behavioral events during the trial. So we've stacked all the trials up here. This is time along the x-axis. This is when the animal initiates the trial. And this is when we present the choice. And the different colors are the phase of the theta oscillation. So you can see that when one of these behavioral events happens, the theta oscillation synchronizes to that event so that the phase now lines up across all of the trials. And you can see that it lasts for several cycles of the oscillation. So we have a measure of how well the theta aligns across trials. And we can see that it only aligns, the alignment only occurs in the theta band. It doesn't occur for other frequencies in the local field potential. And we also only see this alignment when the animal is learning. So it's only when the contingencies are changing and the animal's adjusting its behavior that we see a big increase in this alignment across trials.
Well, why does it start to rise before the stimulus onset? They know the timing. I think in the previous picture we saw it too.
Why does theta start rising before the stimulus onset?
Phase, it looked like in the previous picture too, the phase, I don't know, right before the first dotted line, it looks like we already have some
just here
alignment.
Right. On this one,
You can see it here.
I think it's just smoothing. Yeah. I mean, there is potential for him. It's actually when he fixates that central cue. So there is the potential for the signal to be there in the periphery as well. Yeah. And the value neurons that I showed you earlier, they also phase lock to the theta oscillation. So this is where their peak firing rates occur. And you can see that the peak firing rates are all occurring on the rising phase of the theta oscillation. Now, we're not the only people to have seen this. Plenty of labs have seen various cognitive processes correlating with various measures of the local field potential, various measures of brain oscillations within the frontal lobe. The problem is that it's oftentimes difficult to interpret exactly what those oscillations mean, what exactly their mechanistic function is. One of the problems is we don't know whether they are causally necessary for a cognitive process or whether they are a correlate of the underlying neural firing that's instantiating that cognitive process. Really, what you want to be able to do is you want to be able to causally manipulate your oscillation of interest without affecting other neural oscillations and without affecting the underlying neural firing rates. But that's really challenging because, you know, any of our standard tools in neuroscience, so things like optogenetics or microstimulation or pharmacological manipulations, they're always going to affect everything, right? And so to get around this problem, we developed what we call a closed loop microstimulation protocol, where we basically record the theta oscillation in real time and then use that oscillation as a control signal so that we can apply the pulses of our stimulation to the peak of, well, to any phase of the theta oscillation, but we were actually targeting the peak. And the analogy I like to use is it's like pushing someone on a swing. If you push at random, you're not going to move the swing. You have to time the pushes relative to the oscillation in order to have an effect. So this is what happens when we do this closed loop theta stimulation. This is the number of trials that it takes the animal to adjust its behavior to the changing reward contingencies. Like I said, this is a very easy task for our animals. You can see that they're adjusting their behavior normally in just a handful of trials. But when we do the closed loop theta stimulation, they're really impaired at the task. It's now taking them 100 trials to adjust to these changing reward contingencies. And as a control, we did the exact same thing, but we now used the beta oscillation as a control signal. Now, beta is actually 3 times faster than theta. So when we use it as a control signal, we're actually delivering 3 times as much current into the orbitofrontal cortex. But because it's not synchronized to the theta oscillation, it has 0 effect on the animal's behavior. Okay, so once we got interested in theta, we started getting interested in the hippocampus because that has a very strong theta oscillation. It projects directly to the orbitofrontal cortex, and there are neurons that project to the hippocampus, which we think are the source of one of the main sources of theta in the brain. So we did some simultaneous recordings from the orbitofrontal cortex and the hippocampus. This is just me showing off because we 3D print everything now so that we can target any deep region of the brain using MRI guided trajectories. So the first thing that we saw is that there was a very strong theta oscillation in hippocampus, just as we would expect, but the phase of the oscillation was in synchrony with that in the orbitofrontal cortex, so that they were oscillating in harmony with one another. But the degree of the synchrony varied depending on where the animal was in his learning. So this is the learning, and this is the change in the learning as we change the contingencies. So he's chugging along, we start changing the contingencies, he doesn't notice, his performance starts dropping, but then eventually he notices, and then he starts adjusting his behavior, and then he starts chugging along again. And you can see that this is our measure of phase locking for the orbitofrontal between the 2 areas. And you can see that it disappears when his performance is dropping, but then as he starts adjusting his performance, they actually get very, very strongly in phase with one another before going back to baseline levels. Now, if we do our closed loop microstimulation in the hippocampus, we see the same, it disrupts the theta rhythm, same as in, it disrupts behavior, same as in the orbitofrontal cortex. And so that raises the question for us, what is the hippocampus? What is the hippocampus providing to the orbitofrontal cortex during reward-based learning, which makes it so critical for the animal to learn? So this is what our hippocampal neurons look like across a recording session. So this is all of our trials. They're very, very different from cortical neurons. They're very, very different from the orbitofrontal neurons. And you can see this here. This is a typical orbitofrontal neuron. it's firing away throughout the whole session, right? Because every single trial, it's encoding the value of the pictures. So it's always signaling something. In contrast, our hippocampal neurons are barely ever firing. And then every now and then they'll just suddenly start firing away. So when we saw this, you know, I said to Eric, you're never going to be able to figure out what these neurons are doing. I mean, who knows what happened? Maybe someone slammed a door, maybe the monkey changed position, maybe he heard us talking. Who knows what these random spikes of activity will relate to? And he didn't listen to me because he never does, and that's good. And so he had the idea to take these, remember, we've got 3 pictures, the values are changing across time. And he projected those pictures, he created from those pictures an abstract value space, right? So you can plot them on a single graph where each axis is the value of one of the pictures. And so what each point on this graph indicates is the relative value of the pictures, the relative value of each picture to the other 2 pictures. And because the pictures are changing as we go across the course of the session, there is a trajectory being mapped through this value space. And in fact, these changing pictures over here, they map out a circle in this value space. And we do this twice, right? So the animals are essentially going twice around the circle in this value space. And then we can overlay the activity of the hippocampal neurons onto this trajectory in value space. And when we do that, we find that the neurons are not random at all. They're actually firing to specific locations in this very abstract value space. So remember, there's no actual space in this, right? The animals are simply making choices between pictures of varying values, but because the values are essentially relative to one another, you can think of them as lying somewhere in this space of potential relationships to one another. And in fact, if we look across all of our hippocampal neurons, they have essentially what these are, these value place fields. The purple circles indicate a neuron's place, where a different neuron's place field was. You can see that across all the hippocampal neurons, we essentially tile this entire space, right? So that the animal knows it can use this code to know exactly where it is in this abstract value space. Now, more recently, we've looked to see whether we see evidence of hippocampal replay within this space. Just to briefly remind everyone what that is. You know, place fields are normally measured in rodents, and they usually refer to a physical location in space. So here we have a rodent running along a maze. There are 5 different hippocampal neurons encoding different regions of this space. And then when the animal pauses, we see that the neurons fire a few spikes, and they do so in a sequence that maps out the trajectory that they took through physical space. And this is all work done by Dave Foster's lab. Now we can potentially do the same thing, right? Because we have a trajectory through space. This time it's this abstract value space. Here are our different neurons we recorded on a particular day. And you can see that we map out this trajectory. And then we can look for if we get replay events. And in fact, we do. So the orange arrows are where the animal actually was in the trajectory when these replay events occurred. And essentially, they're encoding future positions around the trajectory. They're firing in sequence, predicting, you know, where the trajectory is going to go through space. So we think that they're related to learning. We see that they tend to occur when the animal receives the reward. And we also notice that the animals actually do better when they go the second time around the circle compared to the first time. And in fact, if we switch the direction on them and make them go around the circle in the opposite direction, they do worse. So they are learning something about this potential trajectory through space. So what we think is going on is basically an idea that was, put forward by Howard Eichenbaum, who argued that the hippocampus was essentially a relational code, and it could use that code for any kind of information. And we typically see spatial information in rodents because that's common to most rodent tasks. And it's a very common, relational feature of our environment. We define things in space relative to one another. But in our task where we're using value, we again have something that's defined relativistically. We define the value of something relative to other things. And the hippocampus now uses exactly the same code for encoding that information. So to summarize then, what we're essentially seeing is that the orbital frontal cortex, we still think that its essential function is assigning value to things in our environment so that we can choose between them optimally. But that's a process that requires an awful lot of different kinds of information. It can include information from the hippocampus, which is encoding the value of things relative to one another, and communicate that information essentially by the two areas synchronizing at the theta oscillation. And then when we go in there with our microstimulation, we're essentially knocking the two areas out of phase with one another and preventing them from communicating that information. You can also see how we might think about building some kind of device that could modify specific behaviors. We could have maladaptive behavior, in this case, a drug addict involved in drug seeking and drug taking behavior. We could identify when that particular outcome is being represented in orbitofrontal activity, and then disrupt that pattern using closed loop theta microstimulation. So I should just finish with what we're doing in terms of future directions. The big thing for us is how many neurons we can record simultaneously. Basically, our decoder performance increases. This is kind of an old slide now because we're way above 20. But it just seems to increase almost linearly as we increase the number of value selective neurons going in. And also only about a third of neurons encode values. So in order to get 20 value selective neurons, you need to record from 60 neurons simultaneously. So this is what we've currently managed. We have 4 32 channel probes. So these probes, they look like a single electrode, but they actually have multiple contacts all the way along them. And this is about 250 micrometer width. So we can lower 4 of these into orbital frontal cortex, 3 into hippocampus. That enables us to record about 300 neurons simultaneously. There are challenges with this. In particular, each of these probes costs about $8,000. So this is about, what, $42,000 worth to record this, this data. This is what we're switching to. This is a project that I have in collaboration with Krishna Shenoy and Tira Moore at Stanford. So this is a NeuroPixels probe. So now we have our same 250 micrometer probe, but now there are 384 contacts etched onto it. It's basically a silicon probe. And this is recording from monkey motor cortex. This is about 300 neurons recorded simultaneously from a single probe. This is what we're hoping to move to. This is a primate pixels probe now. So this now has 4,000 contacts etched onto it. It is 4 centimeters long so that we can get down to orbital frontal cortex. So this is hopefully going to give us 2 orders of magnitude higher than the current probes that we're using. And these are much cheaper as well. They're only about $2,000 each. Unfortunately, we're supposed to get these probes in 2019. They are manufactured by the same nanofabricating plants that fabricate semiconductors, and Ford and Chevrolet can't get semiconductors now. So there's very low chance the Wallis lab is gonna get on that slate. So we've been waiting a long time. Fingers crossed we will have them at the end of this year. Okay, so I just want to thank the people that did this work. Eric, who basically did all the hippocampus work, and Susanna, who did the anterior cingulate work that I presented. And thank you to you for your attention. I wish. Yeah, question over there.
Okay. Yeah, really cool work. I am very interested to see that the decoding and the posterior probability fluctuate, and along tying together with the results that when you take OFC neurons, you can actually predict decode the animal's choices.
Yeah.
Do you think that yeah, the part that contributes the most to decoding the choice is the frequency of oscillating of individual neurons, or like majority of the amateur neurons oscillating towards their final chosen neuron, or along some critical time window, maybe aligned to theta oscillation, some phase locking? That is what contributes to the decoding.
Okay, so I'm going to repeat the question for the Zoom people. So the question was how the flip-flopping relates to, whether the flip-flopping relates to the theta oscillation. We have looked for that. That is an obvious question. We have not found it. So if it's there, it's very subtle. It's not. It's definitely not. Definitely not obvious. The dynamics that we see are very similar to. They are very similar to other areas that people have seen similar sort of flip-flopping, and that has been linked to the theta. So the Mosers saw this flickering in the hippocampus when they were doing this virtual reality where they could sort of teleport an animal from one location to another. And when it got shifted to another location, there was sort of this flickering as it was trying to decide where it was. And that was very similar, that the dynamics of that process were very similar. I guess our working model at the moment is that it's essentially a bistable attractor network in the orbital frontal cortex that where you could think of the depth of the basins in the attractors as proportional to value. And then you'll sort of get a flip-flopping that is not necessarily, that is biased towards the more valuable options. So our best guess is that it's cortical dynamics. We can't rule out oscillations, although it doesn't seem to be driven primarily by them. We also can't rule out top-down influences. So we have another project underway to see whether attention might play a role in, which option is represented. So yeah, those are the directions we've gone.
Sorry, I think it was I badly phrased. I mixed two parts of the question.
Okay.
Together. The first part is so do you think the best contribution of the neurons recording in the OFC that contributes to decoding the right choices? Is it because maybe there are a few neurons that are oscillating more frequently to the final choices, and then that's what contributes to the choice? Because eventually you can actually predict the choices. That means that the oscillation, if you think of the attractor model, like the oscillation, sure, they oscillate, but maybe how frequent they oscillate to, or when they oscillate specific to the decision time is what actually contributes.
Yeah, I think it would be very difficult for us to get that kind of, given the number of neurons that we've got, it would be pretty difficult for us to be able to determine that. Yeah, and the noise, determining the oscillatory dynamics of a single neuron is really tricky because of just how low firing rate and how noisy they are. Yeah, or not, maybe. Are you reading out? Or?
I have my own question,
okay.
But I'm going to be generous to the people on Zoom first.
Okay.
And ask. So the first question, does stimulating at different phases of the theta wave or oscillation make a difference?
It does. So it has to be, the peak is definitely the most effective. Yeah.
But the most effective are always
Most
making it
effective,
worse.
the most effective at making it worse,
How
correct?
about at making it better?
We've not been able to do that. So yeah, this project was originally funded by DARPA, and yes, their aim was to make our brains better or to make the brains of soldiers better. I think the problem is basically that in the normal person, the brain is operating in a is basically operating close to optimal. And so if you do anything to it, you're most likely to disrupt it rather than to help it. You know, if our timing was not, there are lags in the system, and if our timing was not super precise, I think we just end up doing more harm than good. Yeah.
If there are other people behind me asking. So the second question from Rudy wonders if you could spend a few minutes linking the stimulation findings back to the patient who remarried and then took the job 100 miles away.
The stimulation findings. Yeah, I mean, well, it's okay. So the stimulation is essentially disruptive to the patterns of activity that we're seeing. I'm not sure that that's ever going to help out the patients, right? Because they basically don't have an orbital frontal cortex. But in terms of the clinical picture, it's potentially useful. So we know that, for instance, we know that obsessive compulsive patients, when they're ruminating, we see increases activity in orbital frontal cortex. We know that drug addicts, when they're experiencing craving, we see increases of activity in orbital frontal cortex. So if we can detect those patterns associated with those maladaptive behaviors, we potentially have a way to disrupt those patterns, and that in itself could prove useful. I'm going to go to the back because she was first.
I just have a question regarding the hippocampus. I was wondering, did you target different parts of the hippocampus? Because one can wonder that maybe it's different part of the hippocampus that's involved in, you know, like a new choice, like a shuffle, and when you're actually in more steady states. So I was wondering if you like into different parts.
So the question was whether we've looked at different parts of the hippocampus. So yeah, that's a great question. So one of the nice things, so whenever you're doing primate work, you sort of have to justify, why did you not do this in rats? And so I was a little nervous going down into the hippocampus because so much of the work has been done with rats. But it turns out that there's been this big change with primate evolution. So in the rat, the bit that's important is essentially the posterior part of hippocampus in humans. In humans, that's tiny. That's a very small part of the hippocampus, and in monkeys as well. And the anterior part of the hippocampus has gone, which in the rats is quite small, has gotten much bigger, more developed. It's also the part that's implicated in most neuropsychiatric disorders. And not surprisingly, it's the bit that connects with the frontal lobe. So we focused on that, obviously. And then in terms of where we recorded from, we recorded mainly from CA1 and CA3. We didn't see any differences between those two areas. We haven't done like a big mapping of the various fields, but yes. Okay. And Frederick?
The kind of two follow-up questions. That's why I raised my
Okay. All right.
So I'll follow up on the hippocampus. I was kind of intrigued that, I don't know if you mentioned this or you didn't specify it, but the first time you go around that circle of value in your value space,
yep,
you don't really know that these associations are going to be there. So I don't see how you could actually have a place cells that is the relative value of the three things if it's the first time that you're going around the circle. But maybe, but then you mentioned that, oh, it's better when you go the second time around, which makes a lot more sense to me.
Well, no. So if you're so remember the animal is basically estimating the value of the three pictures on every single trial. Right. Right. And it's only changing relatively slowly. Like going the entire way around that circle is like 300 trials or something. Right. So it knows the rough value of the pictures from the past history. Yeah.
And so Is the change constant in terms of the speed? Are you like on a constant speed trajectory? Because
it's constant, but not the same, right? So we have two stable bits within the circle,
right.
and then we go, and then you're going to move around that trajectory to the next stable bit. We vary that speed because that helps us to unconfound it from time. Yeah.
And then when you're in the stable part, do you do the responses? If you have the place value neuron there, do the responses fire all the time, or are they sensitive to the fact that it's changing and predicting?
No, they fire even when it's stable.
When it's stable.
There's no, but they don't. It isn't that there are not more responses there. It's that they don't congregate at the stable parts. But yeah.
Because it seems like it'd be sensitive to the speed of change would be the relative. I think that's most important. If you're trying to predict, I'm learning, right?
Yep.
I'm doing this learning task. I'm really learning that it's changing, not learning that it's particular. I already have the memory that it's a particular value. I'm learning that this is actually the trajectory that's changing.
Yeah, you need both things, right? You need an estimate of the speed, but you also need to know where you're going.
Right.
Yeah.
Right.
Yeah.
Okay. Can I ask a follow-up question?
Sure.
This is from Rudy's question because I was thinking about this too.
Yeah.
When you mentioned about patients, if you don't have an orbital cortex, why are you making the wrong choice? Like because like you're picking like the worst possible things to do instead of just random things.
Well, I guess there's some sampling bias, right? We're only I'm only reporting the crazy things, the bad choices, right? I'm sure there was some random good choices in there as well, but they don't make it into the book.
This case is the same thing. All the stories I hear is always like you become a
yeah, you don't mean
We went from being a boring bookseller to all sorts of interesting things.
Yeah. Yeah, well, that is the challenge, right? It's a challenge to know that whether someone's choices are optimal or suboptimal. The way that we usually get around it is consistency, right? Our choice is temporally consistent, and are they internally consistent? And neither of those is true for an OFC patient. Rich.
So you said 30% of the OFC neurons code value.
Yep.
I don't know if you have thoughts about the other 70% if they're coding value of things that you're not currently studying. Are they doing something different? And kind of related to that is if you can record across days, does say a neuron that kind of monotonic with increasing value, is it always that way, or is it just in a particular context and it might have a very different picture the next day?
Yeah, I don't know that anyone's done it across days, but yeah, I don't know if anybody's done it across days. I know that Paleosciopera has done it across like completely different contexts, and it does seem that if a neuron encodes something and a positive, it tends to do it in the other context as well. So they do seem relatively static. And what was the other question?
Oh, just do you think the other?
Oh, well, the other 70% doing.
Value,
Yeah, no, we do see that. So we see a pretty. There are some neurons that like encode one decision variable, but not another decision variable. So yeah, my guess is that they're in different parts of the value space. Yeah. Okay. Yeah.
So I'm not sure it's entirely clear to me that what your model is about the distinction between the OFC and the hippocampus in the role of value. I mean, you said something that the OFC record values, but it seems to me that it's also relative.
Yeah, no, that's true. It's essentially certainly the hippocampus is there's no dynamics, right? It basically encodes, it fires a bunch of spikes indicating that it's in a particular region of space, and then it's kind of done. Whereas we see we see something that looks much more like a decision-making process taking place in the OFC. So I would say that the hippocampus is contributing to the value predictions, but then the OFC is actually doing the comparison process, right? Yeah. Brooke.
Then going back to that posterior, anterior.
Yeah.
I was wondering if you in your lab were thinking about the anterior hippocampus as having a specific role in value encoding, or if you were hypothesizing there might be like a more abstract gradient or posterior, that's where you have your more place spatial encoding stuff that we see in the rodents, and then anterior regions, you've got more abstract notions like reward or value amongst other things, or is it like values?
Yeah, I don't know. I mean, certainly the anatomy would suggest some kind of functional distinction. I think there's sort of the parahippocampal, more spatial parts do go into the posterior hippocampus preferentially. So my guess is that there has to be some kind of functional organization just because there's anatomical organization. But I mean, I'd be guessing, yeah.
You kind of answered my question, which was you kind of asked about it, just about the role of the hippocampus in the OSC. It seems like the hippocampus might really be playing a central role here. You said the hippocampus is aiding with prediction, but the OSC is still doing the choice. But isn't that choice, influenced by what has been learned in the past? And so that really, that hippocampal information, even though it's not, showing, it's not firing all over the place, kind of showing the process, but it's almost like, I'm going to say this wrong, but it's almost like it's super efficient, like hippocampal fire, boom, that's the thing that you have to pay attention to next time you have those to make your choice.
Yeah, so I think there's sort of an increased appreciation for it. Like Daphne Sohami has some data from hippocampal patients, and she's shown that they're impaired on a lot of the same value-based decision-making tasks that the orbitofrontal patients are impaired on. So yeah, I agree. If you've lost those, if it's a place that's making, a key contribution to the value predictions. Now, I don't think it's the only place, right? I think there's definitely other places involved in, signaling the value of cues in your environment, like the amygdala, for instance, and striatum. But yeah, I think there's becoming an increased appreciation that it does play a role in decision making as well.
I got a Zoom question if I
Okay,
could here
yep.
from Alicia. Does the choice of decoders make a difference in decoding performance?
It did not, no. So we tried a whole bunch of different ones. We definitely did linear decoders. We also did some that were, we also looked in, even went back a step and looked in PCA space. There was, yeah, we must have tried 4 or 5 different decoders because reviewers made us, and it didn't really make a difference. Yeah. Question there. Yep, you.
Yeah, I had a question following up on this theme. I'm wondering what your thought is on if the value that you see encoded in hippocampus has any interaction with what it's traditionally been linked to in terms of spatial navigation and state, like having some state value pair that's computed within that region.
Yeah, so the question is, how does value in the hippocampus relate to the spatial place fields in the hippocampus? It's just very difficult to answer in the monkey, right? Like there are some people who are now looking at spatial place fields in the monkey hippocampus. Dora Angelaki had a paper where she had an animal, running around a room basically while she was recording. And there is some evidence from the rodent literature that the place fields have a relationship to value. So I think that the replay events tend to go towards goal locations, for instance. And there's actually some rodent work come out quite recently, like just the end of last year, showing that. So one of the differences, I've only read this paper once, I need to reread it, but so I may be butchering it. But one of the differences seems to be that hippocampus encodes lots of different spatial locations. It doesn't just encode the reward location, whereas the orbitofrontal cortex tends to locate the goal alone. And so there's some models of how that, theoretical models about that, the relationship between those two things, basically how the hippocampus is encoding sort of the structure of the world, and then the orbital frontal cortex is using that structure to determine the optimal response. Yeah.
It's really cool how these brain areas seem to map onto the trip fusion model. Curious about, and you have the integrator in here, singular cortex. I'm curious about the decision bound variable where, bound is high, you have to accumulate a lot, bound is low, you have to release your decision immediately. I'm wondering how, in your mind, how does that work? Is there another area that says, okay, this decision bound or
well, there's some evidence that it is the anterior singular cortex, I think, that says the decision bound. Some people have seen what looks like an urgency signal. I don't know this literature super well, but I think one of the problems with the accumulator model is that it can get stuck. It can just never ever reach a decision. But obviously we don't get stuck, right? We usually do something, and so there is what's called an urgency signal that basically says if you haven't got to a bound by X, just do something, and that seems to be in anterior. There's evidence that that's in anterior singular cortex. So it would fit, with anterior singular being this sort of control region and encoding a lot of these control parameters.
it would be like a group of neurons and just firing at the same time, like release your movement now or something?
Yeah, exactly. Bob?
Yeah, well, first, I really want to congratulate you because the translational implications and the theoretical implications of this work are just really stunning. So thank you. And I wanted to just respond a little bit to Trevor's question. Yeah, some good things do happen. I don't know if they're really good, but a lot of the OFC patients get OCD symptoms. So one of our cohort became a tremendous expert at bonsai plants, and there's several papers showing emergence of new artistic skills and talents. People have been at overall damage. So it's not. And these are patients who are just like Joni described, that have chaotic lifestyles. But I actually have a question.
Okay.
And the question is a technical one. In some of our work with human orbital intracranial reporting, we don't see really precise anterior, posterior, lateral, medial, tectonic organization. We also see two different tests right next to each other with different, let's go down. But everybody looks at the side orbital tectonics. Do you find anything in your single unit reporting that relates to an area, or is it more distributed along the orbital frontal cortex? So
the question was about how response properties vary within the orbital frontal cortex. We looked for all sorts of organizational schemes, and we never found anything super compelling. So I would say that we're in 11 and 13. If we go more medially than that, the neural responses just get super bad. They're just shitty neurons in the very medial part. If you go more lateral, you start seeing things that look more stimulus driven. There starts being a lot more stimulus information. So you're sort of going into that sort of ventrolateral area that has more sort of attention potential involvement. But yeah, I mean, that's a big chunk of cortical real estate. It's about 10% of the animal's total cortical area, and we can't see any organizational scheme. Yeah.
Thank you.
Rich?
So you made the opening thing that you don't think like DBS can work with like treatment of depression here. And I gather it's because you're saying, it works for movement disorders because it's either like a gain that boosts on the signal, or it's damping down an inhibitory thing like damping down a beta rhythm, and need something more specific for the OFC.
Yep.
But I thought that thinking it that way, Bob's recent comment about that you can really see obsessive behaviors in these individuals. Maybe you think DPS would work for people who are showing like OCD, or because it's going to add noise and muck up. They're not going to get locked into the same decision over and over again. They're going to get more random, which is going to break that repetitive behavior.
Yeah, that's exactly it. But the key is you can't have chronic tonic stimulation. That's not going to do it, right? Like if you're experiencing craving or you're experiencing obsessive rumination, you want it to break that, but then you want it to stop, right? You don't want it to just keep going for your entire life, right? Which is what it's doing in the basal ganglia with Parkinson's patients. So you really just need something that is going to detect when a certain pattern of activity is present, and then switch on the stimulator.
I'd agree that would be better. I guess I still think that, thinking about how you can only disrupt things, right? Presumably you like adding in things when information is maximal, you disrupted behavior, right? With your pulse thing.
Yeah.
Still seems like it should be good for disrupting OCD-like things, even if it's in the chronic stimulation state, just because you're, again, making the person probably more variable in their.
Well, yeah, but then you create literally an OFC patient. It's not clear that that's better.
Just trying to keep less symptoms.
I know, that's what we did with the lobotomies. That was.
I think it's the closed loop approach that you're doing. I mean, I think this week we do the graduate seminar today, the paper from the January, where they basically do closed loop, they have signal in the amygdala that portend in the depressed state, and then they shut it down with the striatum stimulation.
I mean, they're also bringing closed loop into the Parkinson's patients as well.
I'm sure it's better if you could do something more specific, right? So your target is can't do the blasting DBS, can't even do the pulse, but now we got to build up the communication, say, between hippocampus and OFC, or something along those lines, right?
Yeah.
That would be the next stage.
Engineers don't do open loop stimulation circuits. The circuit explodes. They do closed loop.
Was a question by that?
Yeah, kind of curious about different types of worry modality. It seems like you're able to, by disrupting OC, circuits, you're able to disrupt the monkey's ability to learn dissociation between juice. That makes sense to me. Kind of curious about things that are skills that are more natural or inherent, like if the monkey prefers bananas to apples, you disrupt that normal decision-making process. So that seems like it would be more of a connection to a situation where you have a man leading his life for 17 years and people unable to see this in a good color.
Yeah, no, absolutely. People use innate preferences all the time in studying orbital frontal cortex, and you see them represented just as you would see different amounts of juice. So there's definitely no difference. Now, where those innate preferences come from, where they are stored in the brain, who knows, right? I mean, presumably, my best guess would be in the sensory circuits, right? Like the insular cortex, gustatory cortex. Okay.
Okay. Thank you.
Thanks.